Hey there, health enthusiasts! I've got some groundbreaking news that could change the landscape of cancer treatment forever. In December 2024, Russian health authorities made a game-changing announcement: the development of an mRNA-based cancer vaccine named Enteromix. And guess what? Clinical trials are set to begin early this year. Now you might be wondering, what exactly is Enteromix? Well, this therapeutic vaccine is designed specifically for patients who have already been diagnosed with cancer. Its mission? To halt tumor progression and eliminate those nasty metastases that spread cancer throughout the body. This innovative vaccine is the result of collaboration among several top-tier Russian research institutions, including the Gamaleya National Research Center for Epidemiology and Microbiology, the Blokin Cancer Research Center, and the Herzen Research Institute of Oncology. Yep, these are some heavy hitters in the world of medical research.
The term immunotherapy is actually an umbrella term for a number of different modalities that are all designed to use the body's own immune system to help fight cancer. The specifics of each therapy differ, but they all rely on a deep understanding of how the immune system is regulated and how that regulation can be modified to get the desired outcome. It has been known for some time that the immune system plays an important role in monitoring the formation of potentially cancerous cells. It's estimated that an individual cell can have as many as 20,000 DNA-damaging events a day. The vast majority of these are identified and repaired by specific DNA repair pathways. Those cells that do acquire a malignancy that are not repaired are usually removed by the body's immune surveillance system. This generally involves cell-mediated immune mechanisms that rely on differentiating between self and non-self antigens. Often, this DNA damage results in a single-point mutation in a nucleotide that leads to a unique peptide that may be recognized by the immune system as non-self. These tumor-associated antigens are presented on the surface of cells in conjunction with major histocompatibility complex, or MHC molecules. This TAA-MHC complex can then be recognized by a T cell with an appropriate T cell antigen receptor. But in order for the T cell to become activated, a complex mix of co-stimulatory signals are required. One example of this is the CD28, CD80, 86 activation signal. This second signal then activates the T cell to allow it to carry out its effector function. But just as there are signals that activate T cells, there are counteracting signals that can inhibit activation. These inhibitory signals, mediated by cell surface proteins known as checkpoint molecules, normally play an important role in regulating autoimmunity, but their actions can also potentially inhibit a robust immune response to tumors. When a T cell interacts with an antigen-presenting cell, the cell surface marker CTLA4 on the T cell can interact with CD80, 86 on the dendritic cell, causing an inhibitory signal to be sent, essentially down-regulating the T cell's response to the antigen. These inhibitory signals can also be found on some tumor cells. In these cases, the tumor expresses the antigen PDL1, which binds to PD1 on the T cell. This inhibits its activation, even though the T cell receptor has bound the MHC tumor-associated antigen complex. Thus, the tumor cell escapes the immune system surveillance by co-opting checkpoint molecules. But if an antibody to either PD1 or PDL1 were added to the system, it could block the interaction and prevent the inhibitory signal. This is the premise of the class of immunotherapeutic drugs known as checkpoint inhibitors. By preventing the inhibition of the immune response, this new class of drugs allows the immune system to recognize and fight the tumor cells. The inhibitory signals being targeted can come from a number of sources, but they have been extensively characterized in professional antigen-presenting cells and in a subset of T cells known as regulatory T cells. The primary purpose of this T cell subset is to suppress an immune response and maintain homeostasis, preventing autoimmunity. Although the exact mechanisms are still an area of intense study, what is known is that they appear to work through 4 basic modes of action. These include the production of inhibitory cytokines, suppression by cytolysis, suppression by metabolic disruption, and suppression by the modulation of dendritic cell maturation or function. These interactions between the immune system and tumor cells are complex and lie at the heart of immunotherapy. As we begin to understand the molecular basis for these interactions, we can start to see how we can use them to address a disease like cancer.
So how does Enteromix work? It employs mRNA technology, yes, the same technology used in the COVID-19 vaccines by Pfizer and Moderna. This tech stimulates the immune system, training it to target and destroy cancer cells. Kinda like giving your body its own personal army to fight off the invaders. And the best part? Preclinical trials have shown incredibly promising results. We're talking complete disappearance of malignant tumors and metastases in animal models. That's right, complete disappearance. But this isn't just a minor breakthrough, this is revolutionary. But there's more. Enteromix is designed to be personalized for each patient. Using artificial intelligence, scientists can analyze your tumor-specific information and develop a tailored vaccine just for you. And they can do it all within approximately 1 week. Talk about cutting-edge technology. The Russian government is also stepping up in a big way. They plan to provide Enteromix free of charge to patients nationwide. This reflects a significant advancement in cancer treatment and offers new hope to countless individuals and families affected by this devastating disease.